Hello. I thought I would end this unit by talking about how our cells actually show this happening. And of course, by our cells, I also mean animal cells and plant cells and amoebas and bacteria. And so I wanted to give you a concrete example. And then we want to talk about a little bit how cells might regulate this. And that kind of goes back to the beginning of our unit, which when we were talking about my silly analogy of Santa's workshop, right? Not all elves need to be working on all types of toys. You're going to have elves that specialize. Maybe this year the Baby Yoda doll is very popular, so you need to make a lot of them. So, are there cells that only need to make a certain kind of protein? And if so, how do we make that happen? And in fact, we see that. So, I wanted to give you an example, and I titled it A Tale of Two Cells. That's after a pretty famous book, A Tale of Two Cities. So, I'm going to start off with a red blood cell. And then by the end, I'm going to give you the second tale of a red blood cell that has something wrong with it. So let me go ahead and present this so you can see this a little bit bigger. Excuse me. And so what we have here on the bottom, let me explain this a little bit. In all of our bodies, we have cells in our bone marrow that are making new red blood cells. And they are stem cells. They are cells that can just constantly divide. And some of them are going to develop into red blood cells, and others are going to stay stem cells. Now, you do not need to worry about any of these names. That's not my goal. My goal is to show you in this picture, the stem cell is going to change and change and change and change and get smaller. And then suddenly, right around this midpoint, we're going to start seeing the color change. And it's going to end up being a red blood cell. And the color changes because you're going to be making a lot of hemoglobin protein. Now, hemoglobin is a little globular protein, and its goal is to hold oxygen. And a lot of us already know that. In our bloodstream, we have red blood cells that are uh, little bundles of oxygen. They carry four oxygen molecules in each hemoglobin. And these red blood cells have millions of hemoglobin proteins. So they're a perfect example of how this works because in one of these stem cells, right around here, you're going to get a cell that is going to be making a lot of copies of the hemoglobin gene. So in our nucleus, what I did up here in our nucleus is I gave you three potential genes that we find in a human cell. We have one called crystalline, and that's made in our lens of our eyes. We have insulin, which is made in the pancreas. And then we have hemoglobin, which is made in our red blood cells. Now, here's the thing: eye cells, pancreas cells, and red blood cells, the stem cells, they have all of these they have all of these genes. But in the case of the red blood cell, we don't want to make crystalline protein, and we do not want to make insulin. We are not a pancreas cell. We're talking about a red blood cell. In fact, of these three proteins, the only one that the red blood cell wants to make would be hemoglobin. And so notice what I'm drawing here is the DNA double helix, and I'm drawing an mRNA strand being made. And that would be the process of transcription. This is the only gene that's getting transcribed into an mRNA strand. But because this is a red blood cell and we want millions of copies of this, what I drew is just a little sneak peek into this. We're going to see lots and lots and lots of mRNA molecules being transcribed, and then they're going to go out to the cytoplasm. And here I've drawn our little purple ribosome that we did the other day, and these ribosomes are making hemoglobin. And so you can see on this bottom picture we start to make hemoglobin, and we have a huge production of hemoglobin in our red blood cell. But we don't see any insulin and we don't see any crystalline proteins being made. Those genes are silent. They are there, but we're not making anything. Why is that so? How is that? So we kind of sneak back to this idea in our last chapter where cells, as they divide, they can turn into different types of cells. So here's the example, and then here's another one we have, and that would be a nerve cell. So when cells differentiate, they become specialized, and some genes get turned on, and other genes get turned off. Now, when I say on and off, what that means is we actually make what it tells you to make. If you're a pancreas cell, you want to make insulin protein, so your insulin gene needs to be on so you express that gene. But your hemoglobin gene better be turned off. You're a pancreas cell, you don't want to make hemoglobin, you want to make insulin. So notice in our pancreas cell, in this example, we have a glycolysis enzyme gene that's turned on, it's active. We have the crystalline transparent gene that's turned off. We're a pancreas cell, we don't want to make eye proteins. And then the insulin gene is turned on, and then the hemoglobin gene is turned off. If you're a cornea or a lens cell, you're going to be making that eye lens protein, but you want the other ones to be turned off. You want the insulin gene turned off. You're not a pancreas cell, and you're not a red blood cell, so you want the hemoglobin gene turned off. You're not going to make those. But you are a cell that is going through glycolysis, so we want that gene on. And then our nerve cell also has the glycolysis. All cells need to undergo glycolysis. And so you're going to have all cells making that enzyme. So we want the DNA on. So we get this idea that we want to keep some genes silent in some cells and then turn those genes on in other cells. So how does this work? And I'm only going to give you a little sneak peek here into this. How do we regulate what genes get turned on and turned off? Well, it has to do with how we tell the RNA polymerase. Now, if you recall, in one of our previous slides, we learned that RNA polymerase is the enzyme that makes our RNA. Now, it becomes an easy thing then to say if RNA polymerase binds to the DNA and transcribes, well, it's on, it's going to be working. But if we somehow block RNA polymerase, then nothing's going to happen. And so it becomes all about the RNA polymerase. Now, what we need to learn today is that every gene has a specific region called a promoter sequence, and it tends to be upstream, and that simply means in front of the gene. Upstream simply means that it's before the gene, it's upstream from the direction. And we call this a promoter sequence. And RNA polymerase loves the promoter sequence. That's how it kind of latches on, and that's how it starts opening up the DNA to start transcribing. So it's this relationship that we're either going to block or we're going to encourage. So let's take a look at this logic here. If anything blocks the RNA polymerase from starting on the promoter gene, that gene is turned off. It's not expressed. If anything is going to block it, then it's off. If anything is going to attract the RNA polymerase or causes RNA polymerase to attach, then we would say the gene is turned on or it's going to be expressed. So all cells, bacteria and animal and plant and human cells, all cells have promoter regions on all of the DNA, all of the genes. every gene has a promoter region. Now, sometimes cells can do some fun little tricks of grouping genes together with one promoter, but the case still stands that unless that promoter is there, RNA polymerase is not going to bind. And if we block that RNA polymerase from binding, we have, for all intents and purposes, we have blocked, we have turned off that gene. Conversely, if we attract RNA polymerase, if we tell it to come there in some way, that gene is on. So if you are a pancreas cell, you are going to encourage RNA polymerase in binding on the insulin gene, but you're going to somehow block or discourage the hemoglobin gene from being made, and so forth and so on. So that gives us a little bit of a hint as to how some genes get turned on or some genes get turned off. So, what we're going to turn to next is what happens when something goes wrong. And then we're going to wrap up by talking about a specific type of mutation in that hemoglobin gene.